Hi. Female germ cells proliferate by mitosis in the ovaries to form a large number of oogonia. These cells are diploid, containing two X sex chromosomes. They develop into haploid mature oocytes via the process of oogenesis. Now, this process is similar to spermatogenesis, however, there are some significant differences. The ovaries are a pair of organs that produce oocytes and reproductive hormones. They lie near the openings of the uterine tubes, also known as the fallopian tubes or the oviducts, that extend from the uterus. Finger-like projections from the uterine tubes, called the fimbriae, collect oocytes when they are expelled from the ovaries. The oocyte is carried into the uterine tube for fertilization and subsequent implantation into the walls of the uterus. The adult ovary is predominantly made up of connective tissue that supports a large number of follicles. Blood vessels and nerves are concentrated within the central medulla, whereas follicles are found in the outer cortex in varying stages of development. Oogonia begin oogenesis by entering meiosis 1 in week 12 of embryonic development. During meiosis 1, the cell is known as the primary oocyte, and it is surrounded by a thin layer of squamous epithelial cells. The primary oocyte at this stage is developmentally arrested in prophase of meiosis 1. The number of primordial follicles vastly increases during the fetal period, but many degenerate, leaving around 400,000 follicles available at puberty. No new oocytes are formed after birth. Now, with the onset of puberty, some of the primary oocytes continue oogenesis each month. The primary oocyte becomes larger, and the follicular cells around it become cuboidal, and the layer thickens. The follicle is now a primary follicle. The oocyte and the granulosa cells, or the follicle cells, produce a layer of glycoproteins on the surface of the oocytes called the zona pellucida. When a follicle forms more than one layer of granulosa cells, it is called the secondary follicle. One follicle continues to develop and grow, and the others degenerate. A cavity called the antrum forms between the layers of the granulosa cells, and a mass of follicular cells is now termed the cumulus ovorus. The connective tissue cells of this ovary around the follicle respond to differentiation and form two new layers. These layers are the theca interna and the theca externa. The theca interna has a hormonal role, whereas the theca externa has a supportive role. This follicle is now a mature vesicular follicle, also known as a Graafian follicle. The thecal and granulosa cells of the developing follicles produce estrogens that cause the thickening of the endometrial lining of the uterus, as well as other preparations necessary for receiving a fertilized oocyte. This occurs from days 5 to 14 of the menstrual cycle. The primary oocyte of the Graafian follicle responds to surges in follicle-stimulating hormone, or FSH, and luteinizing hormone, or LH, which is produced by the pituitary gland on days 13 and 14 of the menstrual cycle by resuming meiosis 1 and continuing cell division. Now, when the oocyte divides, it forms one large cell and one smaller remnant of the division known as a polar body. At the end of meiosis 1, the oocyte has become a secondary oocyte. Polar bodies are small, non-functional cells. They receive very little of the available cytoplasm and they degenerate soon after division. So the oocyte is able to retain its size but discard chromosomal material to become a haploid cell ready for fertilization. One polar body is formed with meiosis 1 and two polar bodies are formed with meiosis 2. The secondary oocyte begins meiosis 2, but this division is again halted. This time it occurs at metaphase 2. Meiosis 2 will only continue if the oocyte is fertilized. With ovulation, the secondary oocyte is passed into the uterine tube, but the follicle remains within the ovary. At this stage, the follicle is very large and makes up a significant portion of the ovary. This follicle becomes the corpus luteum. In response to luteinizing hormone, the corpus luteum produces progesterone and estrogens and other hormones, causing the endometrium of the uterus to thicken further and develop its vasculature, form glands, as well as prepare for implantation. If fertilization does not occur, the corpus luteum degenerates about 14 days later and becomes a scar tissue remnant of itself called the corpus albicans. Hormone production ceases and menstruation begins as the thickened endometrium is shed. The primary oocyte may be arrested in meiosis 1 throughout life for about 40 to 50 years if it is not triggered to continue development until a menstrual cycle late in reproductive life. DNA fragmentation within those stored oocytes is more common in older women as DNA damage increases with time. This may be the reason for reduced fertility with increasing age. Now, knowledge of the effects of sex hormones on follicle development have allowed invention of the oral contraceptive pill. High levels of estrogens and progesterone inhibit gonadotrophin releasing hormone and subsequently luteinizing hormone and follicle stimulating hormone. Decreased levels of follicle stimulating hormone means that the follicle is not stimulated to develop, and the absence of luteinizing hormone surge prevents ovulation from occurring. Chemotherapy and radiotherapy can destroy primordial ovarian follicles. There is a finite reserve of oocytes formed prenatally, which cannot be replenished after treatment with chemotherapy. Therefore, the preservation of oocytes before treatment begins should be considered. Frozen oocytes may be used for in vitro fertilization at a later date if the patient becomes infertile. Thanks for watching. Please like this video and subscribe to the channel. More videos coming up. See you in the next one.